How's everybody doing?
I'm good. Just don't really enjoy all day in my room.
Well, we're allowed to leave our rooms tomorrow, so that will
I'm
be
not.
nice. Dominic?
Yeah, I'm here permanently.
Oh, you're there permanently. Oh, well. Um,
Unfortunately.
Unfortunately, I'm still free. I'm still free to roam because I don't go anywhere except here, home, and the grocery store. Some people are having technical difficulties this morning for some reason. All right, it's 12:30, although it doesn't look like we have a full slate of people with us today. Um, this weekend lab, what we need to be doing is getting data updated. So I'm going to meet with each group tomorrow. I'll send you an email telling you what time I want to meet with each group. If at all possible, it would be nice to have your data in spreadsheet form for that meeting so that we can assess kind of where you are in data collection and where you're not in data collection. And then we're also going to make a writing assignment for you to begin writing your paper. At this point in theory, you should be capable of starting to write your introduction and your methods, even though you don't have your results yet. You can't start on that, but it's time to begin the writing process. So that will be due in 2 weeks, I believe, to give you time to work on that. We have this week of classes, then we have next week of classes, and then we have Thanksgiving after that. So the process of writing your papers and the process of preparing your oral presentations will be done during that time when we're not meeting in person. But Dr. Judd and I will continue to meet with the groups, probably in most cases individually, to help you through that process, even though we'll be doing it all remotely. There will be a week, and I haven't gotten around to rescheduling, reworking the lab schedule given that we changed projects. But there will be 1 week when we will make a presentation to you so that you can critique kind of what we do in our presentations. It's something that we've done in the past where we get up in front of you and give the kind of presentation that we're expecting you to do so you can see the kinds of strategies and things that we use to communicate effectively. We'll actually give you a comment sheet where you can go on there and kind of evaluate what you liked about what we were doing, what you didn't like about what we were doing, what you found confusing, what was clarifying, et cetera, et cetera. Learning how to make an effective oral presentation is just like learning how to effectively write. You do it by seeing other people doing things right. You learn to write by reading good writing, and you learn to write by practicing writing. In the same way, you learn to present well by seeing other people present well, and you learn to present well by practicing the act of presenting. So we'll be doing some activities with you in lab that will hopefully get you down those paths. you want to say anything about that, Dr. Judd?
No, I just want to add that for those of you on Thursday, I will also be meeting with you by group. So make sure you have things in that spreadsheet or Excel form we've talked about.
Yeah, in all cases, your Excel spreadsheets should take the form of the data sheets that you've been recording data on manually. One of the things that you should always do when you have a project, like any project, but like the ones that you're working on in class, is you should collect data in a way that then makes it intuitively obvious how that data will be analyzed. And if you can think through that enough in advance, you can create data sheets that are easily transferable to an Excel spreadsheet that is then easily amenable to doing an analysis of those data. And I think, Nathan, we did a pretty decent job of that, didn't we?
I think so. And then I also have been trying to, I had a little bit of carryover from the ecology class I'm teaching where we talked about long format versus wide format data, which is related to how we presented those spreadsheets that you all have been writing things down on.
Okay. We have your literature summaries graded. Dr. Judd has class right after this, but after he's out of class, he'll work on getting his that he graded back to you. I don't have class between now and the 3 o'clock hour, so expect to start getting emails from me. I'm going to take a picture of the rubric, take an image of the rubric that we use to grade, and that'll be attached along with your graded summary. Read the comments both in the rubric and on the summary. In some cases, we'll write quite a lot on your summary, and it's good to read through those things. The other thing is that we will sometimes write some summary comments on the rubric itself that don't necessarily appear on the manuscript. So read both of those. My impression was that some people didn't fully understand the paper, and that negatively affected their performance on the literature summary. Some people understood the summary, but then didn't do a great job of doing forward-thinking critical thought about the paper. And then some people did all those things, and that kind of separated the class into sort of 3 sort of grade categories, broadly speaking. As we approach the Veen et al. Summary, make sure that you understand what the paper is about and what the paper is concluding. Hopefully those things have been becoming more clear as we've gotten further into the Konvic Prairie case study. But if those things seem unclear to you, by all means, seek out Dr. Judd or myself. A number of you have been by to see both Dr. Judd in our offices to ask questions either about exams, about study habits, about the literature summaries, et cetera, et cetera. Talk to your classmates and ask them if coming to CS has been productive in preparing for those things or not. And I hope that what they will say is that it was productive, because that's our goal ultimately. But once again, we can't know that you're not understanding something if you don't let us know that you're not understanding something. So feel free to come by and get help on the things that you need help for.
I would just add 2 things. When you're asking yourself the question, did I understand the paper? Did I understand what specifically it's about? Part of your answer to that should include an understanding of the experimental design. So a lot of you understood what happened at Academy Bay, but didn't necessarily explain why what was observed at El Garapatero was informative compared to what happened at Academy Bay. And similarly with the Veen et al. Paper, we're going to have an even more sophisticated experimental design to think about. Several different combinations of variables, several different treatments, groups. And you'll need to be able to think about why each one of those is informative in light of the others. And then the little thing is, remember to format your species names correctly. Italics, lowercase epithet.
So on that front about experimental design, I'm going to share my screen with you because I'm going to be doing it eventually anyway. So there is a PowerPoint that we have for the discussion that is going to be on Friday. Can you guys see this PowerPoint? Okay.
Yes.
So when we went through the ANOVA Well, now it's not sharing. You know, I really don't like Zoom. I like Zoom better. I like Zoom better in the past, but for some reason, when I go to share, sometimes it just doesn't do it. Okay, so when we went through the ANOVA, I gave you this way of thinking about the experimental design of the Vian et al. Paper. You have 1-year burns and you have 4-year burns. In all of these watersheds, you have bison. So bison are grazing everywhere. And so the way you get ungrazed plots is you put up what they call exclosures. These are like enclosures, except trying to keep something in, you're trying to keep something out. And the something that you're trying to keep out is the bison. So each of these green squares represents a place where there's no grazing. Everywhere outside these exclosures is where there's grazing allowed. And then they measured plant communities and nutrients and things like this, both inside these ungrazed exclosures and outside in these grazed areas. And so here you have their whole design. You have 1-year burns versus 4-year burns, and then you have ungrazed areas in the green and grazed areas in the red or pink. This is the way I presented it to you when we went through it in the ANOVA presentation. But another way of thinking about it is to think of it as you have 2 main effects, a grazing effect and a burn frequency effect. The grazing effect is you either have an ungrazed area where there aren't bison or a grazed area. Remember, the ungrazed area is inside these exclosures. And then the burn, so that's then represented graphically by bison there. And then you have 1-year burn frequency or 4-year burn frequency. You can think of that as a lot of fire or less fire. And so you can test in this experiment the effect of ungrazed and grazed by looking at these 2 categories versus these 2 categories. You can look at 1 year and 4, you can look at the effect of burn frequency by looking at this category, 1-year burn frequency versus this category. But you can look at these 2 things as they interact with one another by looking at, for example, does ungrazed 1-year frequency do something different than ungrazed 4-year burn frequency relative to those 2 treatments, 1-year and 4-year burns, in grazed areas. So it's kind of like not looking at column or row, but looking across diagonals at the effect of, for example, ungrazed 1-year versus grazed 4-year burns. And so that when they talk about interaction effects, that's what they're talking about. Another thing, and I was talking to, who was I talking to earlier today? Kat, right? Is Kat here? Anyway, one of the other things that I try and do is I try and say for any given figure, which of the hypotheses is this figure addressing? And when we go through this, you'll see that I've annotated all of the figures with which hypotheses are these figures actually addressing. And it's just my way of kind of systematically pulling apart the figures to figure out what are these figures meaning relative to the hypotheses. And it's a good way of kind of pulling the figures apart to get out what they're actually saying about the hypotheses. And you'll see that I do this on every one of the figures.
Can you go back to that annotated figure? This is a case that I've noticed several times in other classes where people maybe don't carefully read the figure caption. Something that I've noticed people miss, even when they do read the caption, but maybe not as carefully as they could, is that the different letters above the bars indicate significant differences. So make sure you understand what that means when you say, for example, C and C in the upper right corner there. It looks like the gray bar is higher than the black bar in the upper right corner here, but is that difference significant or not?
Is that a question you want them to answer?
Yeah, to anybody.
Not significant.
It's not significant because they have the same letter, correct? So you can't say there's a difference. There's not a significant difference between the species richness in grazed 4-year burn or grazed 1-year burn. By contrast, where you see the mouse now, A and B, is that difference significant? Yeah, I see a lot of nods. How would you say that in a complete sentence? What's going on with those two bars? Sorry, the y-axis is diversity, not species richness. I think I said the wrong one. But how would you explain the black bar and the gray bar where the mouse is in a complete sentence? Anybody want to take a stab at it? You can get help from a friend if you need it.
Remember what the black and the gray bars are, first of all. So once again, reading the caption carefully is important. What's the difference between the black bar and the gray bar in terms of what they represent?
Fire frequency.
Okay, so 1 year burn versus 4 year burn. And so what's going on with these two bars here? This is in which? Grazed or ungrazed?
Ungrazed.
Okay, so take it, Riley.
It's showing that there is a difference between ungrazed fire frequency of 1 year and 4 years.
Is it just a difference or is that difference directional?
Directional?
Yeah, so you could be actually more specific, not just say that there's a difference, but 4 year burns are what relative to 1 year burns in ungrazed areas?
Sorry, I gotta find which one the 4 year burn is.
4 year burn is gray.
So the diversity is higher with the 4 year burn ungrazed.
Yeah,
Nice.
exactly. So
That's what we're looking for. Good.
yeah, so 1 year burns ungrazed is lower than 4 year burns grazed. And what are those two relative to all of the grazed areas?
Somebody
In
else?
terms of diversity?
Are you looking for the fact that they're lower than
They're,
the grazed?
that is exactly what I'm looking for. So the grazed area has higher diversity regardless of burn frequency, and burn frequency doesn't really, or burn frequency only matters in the ungrazed areas. And that's the nature of an interaction. When the burn frequency has no effect in one place and has an effect in another condition, so no effect in the grazed areas, but there is an effect in the ungrazed area, that's the nature of an interaction. In the same way here, the response in terms of dominance to grazed and ungrazed, those things are responding differently. 1 year and 4 year burns are responding differently in ungrazed than they are in grazed. That also represents an interaction. So hopefully this will help you interpret the figures a little bit. Any questions about that? So let me follow that up, just your understanding. Which of these plots does not demonstrate an interaction? Nathan is smiling, because that was a good follow-up question.
It's a good question, yeah.
Even if I do say so myself. Which of these plots does not have an interaction evident in it?
Is it A?
It is a why. Who was that? Riley. Somebody other than Riley explain why Riley is absolutely correct.
I mean, I would guess because it has A, and B. There's no different letters maybe there.
That is exactly it. The response in terms of species richness, the response of 1 year and 4 year burns is the same in grazed as it is in ungrazed. They're just different between grazed and ungrazed. They don't vary among burn frequency treatments, even though they do vary among grazing treatments.
If I gave you a plot that had about 15 species in it, would you know whether it was burned every 4 years or every year? would you know whether it's grazed or ungrazed? If there were 15 species?
He's asking you to look at the y-axis.
Yeah. Who was that? You were right. Grazed. Yeah, cat. Right. But would you know whether it was burned every 4 years or every 1 year?
No.
Right. You wouldn't. Good.
Okay. All right, cool. All right, well, hopefully that helps you prepare for Friday. All right, back to nutrient cycling. I left you guys with a task. Oh, darn it. Why can I not get my mouse to go over? All right. Asked you guys, so can you guys see all the chemical formulas on the screen? Okay. Hunter, which one of these is a carbohydrate?
Top left.
Top left, okay. Grant, which one of these is a nucleic acid? Grant, are you there? Grant is not there.
Is
Sydney.
it the
Oh, Grant, there you are.
the bottom right?
It is the bottom right. How do you know that it's the bottom right?
Because they have the hexagon shapes attached to the pentagon shapes. At least that's
Okay.
how right there.
These right here?
Yeah.
Okay, so those are nitrogenous bases, but not all nitrogenous bases have that structure, because some of the nitrogenous bases are single ring structures. What do you have here?
It looks like a phosphate.
Phosphate bonded to, what is this? This goes back to material from the last test, right?
Yeah.
Hate it when that happens.
Is
help Grant out.
Is that a ribose?
Yeah, that is a ribose. Yep. So it's got a phosphate sugar, phosphate sugar, phosphate sugar backbone with these nitrogenous bases in between. So somebody other than Grant, let's take Sydney. Is this DNA or is this RNA?
Would it be DNA?
Why would it be DNA?
Is that because it only has like a single deoxyribose like bond?
Okay, and I made a mistake. This is not ribose. This is actually deoxyribose. How can we tell DNA from RNA? What's the fundamental difference between the two of them?
DNA has like the double helix, but then RNA is just a single strand.
Yeah, so this is one strand bonded through hydrogen bonding to a second strand. Good. Where up here is a protein, or at least the subunits of a protein?
Bottom left.
Okay, bottom left. Why would you think that that's a protein, Tessa?
Oh wait, I'm so sorry. That's a lipid, right?
That is a lipid. Okay, so that's a lipid. Why? Okay, so tell me, why do you think it's a lipid?
Because it has the 3 chains.
Okay, so it's got a glycerol molecule here. It's got these carboxyl groups, and attached to those carboxyl groups are these long carbon chains. So that means somebody else. Where's the amino acid?
Top right.
Top right. Good, Ryan. Why do you say that's an amino acid?
I'm not sure why. I just know that that's the one.
Okay, so for one thing, I made a mistake. I thought I had gotten rid of the thing that said peptide bond. Peptide bonds are things that join amino acids together. But within this amino acid, what is this thing called? Somebody other than Ryan, since Ryan's having a little bit of difficulty with this one. What is this group called? Or Ryan, if you know what this is called, jump in.
Is it a hydrogen bond?
This isn't a hydrogen bond. It's a special type of section of this whole thing. So this whole thing is the amino acid, and it has on one end an amine group, and on the other end it has a carboxyl group, and then it has this single carbon in the middle that then has a side chain attached to it. In this case, the side chain is just a hydrogen, but it could have a variety of different side chains. So amino acids have an amino group and a carboxyl group within a central carbon, and then those get bonded together from one amino group to the next carboxyl, and that's what makes the peptide bond. What is this thing in the middle here?
Is it a phospholipid?
That's a phospholipid, Sophia. So tell me what you know about phospholipids.
Well, it has the tails like from the lipids, and then it has a phosphate group.
Yeah, so instead of so it has the fatty acid chains, and then it's got the glycerol molecule, but then instead of this glycerol molecule being attached to another fatty acid chain, it's actually attached to a phosphate group. And these things are used in cell membranes. So the phospholipid bilayer is made up of these things. So we have two lipids, a carbohydrate, a amino acid, and then a nucleic acid. And then nucleic acids are actually made up of three different kinds of compounds: nitrogenous bases, sugars, and phosphate molecules. So when we think about this, these are like the major building blocks of organisms. So we can think about carbohydrates, we can think about lipids, we can think about proteins, and we can think about nucleic acids, and we can think about these things in terms of what are the various elements that these different major classes of biological macromolecules require. And of course, what you see in these figures is all of these require carbon, hydrogen, and oxygen. But what are the things that separate these in terms of other elements that they need? Carbohydrates generally are just what? Relative to elemental composition.
Carbon, hydrogen, and oxygen.
Yeah, that's all they really have in them. What about lipids?
They need a backbone.
Yeah, but what's that made up of?
Oh, like mostly carbon, like carbon chains.
Carbon chains, but there's also a carboxyl group in there as well.
Oh, right.
Those carboxyl groups have oxygen, and those carbon chains have a bunch of hydrogens attached to them. But then what else do some lipids have that other lipids don't have?
Nitrogen.
Who was that?
Dominic.
Dominic, is there nitrogen?
Yes.
Look at the middle one.
Phosphate.
Oh, no. Yeah, phosphate.
Phosphorus. Phosphorus is in there. My clipboard keeps sliding down.
That one happens to have an N in it as well, but the key
Oh
there
yeah,
is
it
that
does
you've
have
got
an additional thing in there.
But notice the
Oh.
phosphate is labeled, right? So now you got it, Dominic.
Yeah. Okay. Cool. Yeah, I didn't notice that. I'm too far away from my screen. Oh yeah. Okay. What about proteins?
They also have nitrogen.
They also have nitrogen, and if you go and look at a table full of amino acids, what else? The things on the right are amino acids. What else do you need sometimes? This is actually look at cysteine.
Is it sulfur?
It is sulfur. And that brings us to nucleic acids. What elements do we need for nucleic acids?
Nitrogen and phosphorus?
We need nitrogen and phosphorus. So by looking at what the major biological macromolecules are, we can figure out what kinds of elements an ecologist might want to pay attention to as we think about nutrient cycling. And that doesn't mean that these are the only things that we need to think about, because there are all sorts of trace elements that are important in physiological function. For example, for animals, calcium is super important for the function of muscles. Sodium is super important for, sodium and potassium are super important for the function of nerves. Potassium is important in plants, calcium is important in plants. So there are these other trace elements that are also important, but at least for the big things that make up most of the dry mass of an organism, these are the things that we need to think about. And what we're gonna do today is we're gonna concentrate on carbon and nitrogen, because carbon is probably the simplest, one of the simplest of all of the biogeochemical cycles that we need to think about, and nitrogen is probably one of the most complicated. So we're going to give you two ends of the spectrum, but if you can understand how those work, it gives you some insight as to how any of these things would work. So that's kind of the goal today. Questions so far? All right. Dr. Judd has told you about ecosystems. So we think of an ecosystem as all the organisms in an area and all the abiotic materials and energy that they interact with. And so when we start thinking about this from the standpoint of nutrient cycling, we can think of nutrients as being in certain places at any given time. And we can think of the nutrients either being in pools or in fluxes. And a pool is just a place where a quantity stays for a particular period of time. And so Dr. Judd is a pool of nitrogen. I am a pool of nitrogen. Dominic is a pool of nitrogen. When any of us dies, especially if we die out in a forest and nobody finds our body because we have no friends, we start to decompose, and all of that nitrogen that is in our bodies now goes into the soil. Well, that movement of nitrogen out of our bodies and into the soil, that represents a flux or a flow from one pool to another pool. Dominic and Nathan, I hope you guys don't die anytime soon. You were just an example. So when you think of cycling of these things, think of things as either kind of residing in places for long periods of time in many cases, or moving from one of these long-term storage areas to another long-term storage area. And then the cycle is just the collection of all of the major pools and fluxes that you might think about. So we can think about this from the standpoint of the nitrogen cycle, or the carbon cycle, or the phosphorus cycle, or the sulfur cycle. But basically, we have pool 1, we have pool 2, we have one flux that moves from pool 1 to pool 2, and a second flux that goes from pool 2 to pool 1. This is not a very realistic cycle, but we can make these cycles more complicated where you have lots of fluxes and lots of pools. And we can also think about spatially where these pools are located. Are the pools on the surface of the ground? Are they down in the soil? Are they the atmosphere? And these fluxes can be classified as either inputs or transformations or outputs. Here, if this is an ecosystem, and let's say pool 1 is the atmosphere, the movement from the atmosphere to the ecosystem might be viewed as an input. The movement from pool 4 out to pool 5, let's say that pool 5 is stream flow or groundwater or something like that, that might be the loss of a nutrient from the ecosystem to stream flow, for example, which then carries it away from the ecosystem. That would be an output. Flux 2 is a movement from pool 2 to pool 3. Well, that flux could actually be due to the nutrient moving from pool 2 to pool 3 in its current form and it doesn't undergo a change at all, or it could actually change form as you move from pool 2 to pool 3, and we would refer to that as a transformation. So glucose going from glucose to carbon dioxide during respiration, that would be a transformation, whereas glucose moving from one organism to another organism through predation would just be a flux without any transformation. Is that all clear to you? All right. So let's think about the carbon cycle. And I want to start with carbon cycle because Dr. Judd just got finished talking to you about the two major processes that are involved in the carbon cycle, at least one of them. In this figure, which one of these is cellular respiration and which of these is photosynthesis?
Cell respiration is the top one, right?
Cellular respiration is not the top one. Why is it not the top one? Was that Dominic?
Yes.
Okay, what are your starting points? Think of the
Is it because of the
reactants that are on the left-hand side. What's that?
Is it because of the chlorophyll?
So one clue is chlorophyll, but another clue is think about the reactants on the left side and the products on the right side. What is this product right here?
Glucose.
This is glucose. So because glucose is one of the products of this particular equation, that should clue you in that this is photosynthesis because that's how we make glucose. And what do we start with, Dominic, when we are making glucose? From your knowledge of what Dr. Judd went through with you last week. This happens at the beginning of the Calvin cycle. Something gets added to something else using Rubisco. Somebody help Dominic out. Connor, haven't heard from you in a while.
I can't help him out.
Oh, okay. Call in somebody on your screen to help you out.
Audrey, you want to help me out?
Doesn't Rubisco use oxygen?
Rubisco can use oxygen, but when you're going through photosynthesis to produce glucose, what is Rubisco going to be binding with?
Is it the carbon?
It is the carbon. Was that Dominic again?
Yeah.
Yeah, there you go. Carbon dioxide. So we take the carbon dioxide, we get enough of that carbon dioxide, go through enough cycles of the Calvin cycle that we build up a molecule of glucose. We use the enzyme Rubisco. So there are lots of clues in this equation to show us that this is photosynthesis. And photosynthesis is the process of going from these low energy compounds, carbon dioxide and water, to a higher energy compound, glucose. Cellular respiration, on the other hand, takes this high energy compound, glucose, breaks it down to release energy, which we often see in the form of heat, and the byproducts of that reaction are the liberation of carbon dioxide and the liberation of water. And so you can think of these two equations as kind of going in opposite directions. Photosynthesis requires light energy. Why does photosynthesis require energy input? Trying to build on what Dr. Judd taught you last week. Connor, since you couldn't help us out last time, help us out now. What's up, Connor?
I don't know.
Why do we need energy to take this carbon dioxide and water and make glucose?
Don't we need the energy in order for the CO2 to bond to the Rubisco?
So let's see, is that what we mainly need the energy for? It's not really what we mainly need the energy for. Yeah, go ahead, Nathan.
Yeah, so the energy comes into the system as light, right? As photons. How is that energy stored?
Are you talking about light reactions or the Kelvin cycle, Nathan?
Well, I'm asking them about if you've got energy that's existing as light and you want to get it into like chemical energy was what you're going for, right?
Yep.
Yeah.
Glucose
so
is the long-term storage form of energy.
Well, now I just walked around my own question. But to be, because glucose is an energy-dense molecule and carbon dioxide is not so energy dense, getting from carbon dioxide to glucose requires a lot of energy. The function of Rubisco is to make is to reduce the amount of effort necessary to do that. So such that you can go from carbon dioxide to sugar with just light instead of even more energy than you would need if you didn't have an enzyme to help out. I'll go ahead with the diagram.
So this is carbon dioxide, carbon double bonded to 2 oxygens. Each oxygen has a double bond because carbon supports 4 bonds. Glucose, on the other hand, let's see.
Yeah.
Now, is this the right arrangement of it, Nathan? I didn't
I'm
prepare
checking.
to do this.
Yeah, you've got the ring of carbons and then the upper
oxygen.
left. Yeah, oxygen. Yeah.
Carbon coming off here, carbon coming off here. Carbon coming off here.
Where's your O?
O is right here.
Yes, adjacent to the O. Yep.
Yep, okay. So here we have 1, 2, 3, 4, 5, 6 carbons all bonded to one another. How does a chloroplast facilitate that? You now have 6 bonds between all these carbon molecules. You've taken in 6 molecules of carbon dioxide to build this glucose molecule. Why do we need energy in order to do this? Why is this a higher energy molecule than this one is?
Because it has more bonds.
Okay, it's simply a factor of all of the bonds holding that thing together. And so to go from these low energy molecules that are small, don't have a lot of bond energy contained in them, to this higher energy molecule with a lot of bonds in them, you have to input energy into the system. That energy then gets stored in that long-term storage molecule, glucose. And then when another organism consumes that glucose and breaks down that glucose into its smaller low energy components, all of the energy and all of those bonds gets released. And that's the energy that you use to do whatever work you need to be doing inside the cells of your body. And so you can think of these two things as being an energy intensive process of going from low energy molecules to high energy storage molecules, or the opposite reaction, which takes those high energy molecules and releases the energy that is pent up in all of those bonds between those carbon those carbon atoms. So one of the nice things about the carbon cycle is these are the two big transformations of carbon. Transformation of carbon from carbon dioxide to glucose, and then the transformation of carbon from glucose to carbon dioxide through the process of photosynthesis and respiration. So one of our tasks is to start thinking about an ecosystem. And the way I'm going to illustrate this ecosystem is by basically building a big box that represents soil. Now, what kind of organisms live on top of the soil?
Plants.
Okay, so we can have plants. So plants can live on top of the soil, but do plants only live on top of the soil? Daniel, you're nodding your head yes?
Yes.
Where else do they live though?
Other animals?
Under
Well,
the
no,
soil.
I'm talking plants now.
Dominic has something to add.
Look at this plant here. Dominic, do you have something to add?
Under the soil.
Yeah, they also have roots. So we could think of this as the shoot, the above ground biomass part of the plant, and then we have the root down here, which is the below ground biomass part of the plant. So plants live in both places within the same individual. Part of an individual is above ground, part of an individual is below ground. Somebody mentioned animals. Is that Daniel?
Yeah, that was me. I can't.
So
find out more.
one of the things we could do is we could think of animals as living on top of the ground. We could also have animals in the soil. By all means, there are plenty of animals that live in the soil. What other kinds of organisms might we want to think about?
Does like algae or fungi count?
Sure. So we could put fungi. Do fungi live on top of the ground or below the ground?
On top.
Okay, well, where else do they live?
Low.
What's that, Keaton?
Low.
Where is most of the biomass of a fungus? Above ground or below ground? Kat?
Below.
Most of the fungus is below ground. So we could actually represent things relative to their sizes. So each of these things refers to a pool. We have a pool of animals, we have a pool of fungi, we have a pool of plants. What are some other pools that we can think about from the standpoint of carbon? We have a pool of soil, we have a pool of animals, pool of plants, pools of fungi. What's another big pool that we need to think about, especially from the standpoint of photosynthesis?
Bacteria.
Bacteria, can we not talk about bacteria? I'm really more of a big picture guy. But no, bacteria, certainly. We can have bacteria on top of the soil. We can have bacteria within the soil. We can have bacteria on animals. We could have bacteria on fungi. We'd have bacteria on plants. Basically, bacteria are everywhere.
Bacteria are one of those things that we tend to explore their carbon cycling role at smaller scales, and we just kind of let it all average out at big scales like this.
Yeah, one of my former students teaches at the University of Montana, and he actually studies how differences in the types of species that live in soil affect rates of soil respiration, which we'll talk about here in a little while.
Where else can you find? Oh, go ahead.
He has to do a lot of sequencing of soil. He runs DNA sequencers that just look at all of the different DNA sequences that are in soil to try and figure out what microbes are there. Go ahead, Nathan.
But where else can you find carbon? Where else is carbon on Earth besides in the soil, in the plants, and in the animals?
In the atmosphere.
Thank you very much. Yep. So there's this atmospheric pool. So now we have kind of all of the major pools identified. We have soil as an aggregate. Within the soil, we have roots of plants. We have the mycelia of fungi. We have animals that live in the soil. We have bacteria that live in the soil. Now we can start thinking about fluxes. What are some major important fluxes that we need to think about?
Between carbon and the plants.
Okay, not carbon in the plants, but between
plants and carbon. Oh, sure. Between.
Well, this is all carbon moving around. So carbon is going to be moving.
Do you mean like the flux between like atmospheric carbon and carbon in plants?
Sure, that would be a flux. What would we label that flux as?
Photosynthesis?
Yeah, this would be photosynthesis. So is this flux then just a simple flux, or is this a transformation?
It would be a transformation, right?
Why?
Because it's turning that carbon into glucose?
So up in here in the atmosphere, we can say that this is carbon dioxide, and we can say that this down here is glucose. So for each of these pools, we can not only identify what the pool is, but we can also identify the form of the matter that is in the pool. The form of carbon that is in the atmosphere is carbon dioxide. The form of matter that is in plants is glucose or some derivative of glucose. Cellulose is just a bunch of glucose molecules bonded to one another in a particular way. And so we can think of glucose in plants, carbon in plants as primarily glucose. In terms of dry weight, I'm guessing, Nathan, most of plants is glucose, right? Can't hear you.
Oh, yes, packaged as cellulose and starch. You'll often see people talk about organic carbon once you turn it into an organism like a plant or an animal.
Yeah. So what would be another flux that we could put up here?
Human respiration or animal respiration.
Okay, so respiration. Respiration is organisms that are animals going through cellular respiration, metabolism, taking what form of carbon in animals?
What was the question? Sorry.
What form of carbon is in animals?
Oh, I don't know that one.
From an energy standpoint. I can't tell who's trying to talk.
Is it carbon dioxide?
Not carbon dioxide in the animals. Carbon dioxide is going to be in the atmosphere because we're breaking down what to release carbon dioxide so animals can get energy from it. That was Joseph, right?
Is it oxygen?
Not oxygen, we're talking about carbon here. So everything should be carbon containing.
Where does the carbon dioxide that animals exhale come from?
Well, we inhale oxygen, right? And then we go through like the process of respiration and output CO2. I guess we talked.
But where does the carbon come from? If the oxygen is oxygen molecules, where do those carbon atoms come from that are in the CO2?
Don't confuse respiration with breathing. I try and usually be very clear about this. Breathing is us breathing in and out. When we use the term respiration in biology and in biochemistry, we're generally referring to the second equation down here, which is cellular respiration, which is breaking down what and releasing energy and carbon dioxide.
Is that the glucose?
That is glucose. So we're going to have glucose here, C6H12O6 here. This then is cellular respiration. Taking glucose and converting it to carbon dioxide, in the process releasing energy that the animals use to do whatever animals need energy for. Are animals the only things that do cellular respiration? Tessa, who does cellular respiration?
Plants and bacteria, and I think fungi as well.
That's also cellular respiration from all of those different organisms in all of those different pools within this ecosystem. All of those arrows would be labeled cellular respiration, breaking down glucose and returning it to the atmosphere. Now, bacteria breaking down glucose in soils and returning it to the atmosphere has a special name. It's called soil respiration. And so we oftentimes think of that as different from all of these other forms of respiration, but they're all still the same chemical process going on. What am I leaving out, Dr. Judd?
Yeah, I'm looking. I just maximized your screen.
What happened when
right.
Dr. Judd and Dominic died in the forest? and nobody found their bodies for a while because they have no friends.
They decompose.
Yeah. So all of these things that live on the surface and underground, when they die, they go through the process of decomposition. So that's another flux. But in this case, so it actually has two processes. One process is death, in which case you go from living organic matter to dead organic matter. But then when you're dead organic matter in the soil, bacteria will attack your body and decompose you. And that process of decomposition is actually just this same equation, taking the glucose that is in that dead body. The bacteria bring in that glucose, they break that glucose down to get energy from it, and they release that energy to the atmosphere. They release the byproducts of that chemical reaction to the atmosphere in the form of CO2. So this movement of animals to soil is death, and then the process of those things breaking down then is decomposition. And so it's not difficult to think about an ecosystem in terms of pools of where these things reside, and then in terms of fluxes, how material moves from one pool to another.
One last question for everybody. There's all that glucose in the animals that they're respiring into the atmosphere. Where did the carbon in the animals come from?
Let me get rid of some of these fluxes so we can see things a little better.
The animals started out as little babies or eggs or whatever they were, and then they became large mature organisms. Where did that carbon come from?
Where does glucose come from?
What they eat.
From the food.
Yeah, what they eat.
So these guys are eating plants. They're maybe eating fungi. They may actually be eating bacteria. All of those things, that consumption, in this case, heterotrophic consumption, is basically moving glucose from one organism to another organism. Once again, this is not a transformation, it's just a flux, a simple flux. Taking the glucose in fungi, bringing it into animals, taking the glucose in plants and bringing it into animals. For that matter, fungi can do the same thing with plants in reality. Bacteria can get energy from plants. We've talked about bacteria. Have we talked about bacteria getting energy from plants? We haven't yet. We will, though, in more detail. So yeah, thanks. I almost forgot that one, Nathan. So we can take all of this and we can put it in a cleaner, although also more complicated sort of nutrient cycling diagram. And you'll also see in this diagram that there are some things that we didn't mention. What are some things that we didn't mention in our simplified version that are in this more complicated version?
Fossil fuel.
Okay, so there are fossil fuel deposits. So one of the things that happens over geological time, who was that? Was that Brandon? Oh, that was Dominic. Okay. So we have fossil fuels. When organisms die, their bodies decompose. Some of that decomposed carbon gets pent up in soil. And if that soil eventually becomes rock, it can undergo pressure and heat and ultimately be converted into fossil fuels. And so deep in the ground, there are fossil fuel deposits, and humans dig up those fossil fuels and burn them. So there's also this fossil fuel combustion that takes essentially hydrocarbons, not glucose, but used to be glucose, and burns them off and creates CO2 in the process of releasing that energy in oil and gas and natural gas deposits. What else is in this figure that's not in the figure that we had before? We've got respiration, we've got soil respiration, we've got death. What else do we have?
Excretion.
Excretion. So when bison defecates on the landscape, there's nitrogen and phosphorus and stuff in there, but there's also carbon in there as well. And so excretion is important. What else is in this figure that wasn't in the figure that I wrote on my board?
Groundwater? Is that
Okay, so
significant?
groundwater isn't really involved in here because there's no fluxes going from, so for example, soil to groundwater. And that's actually not really all that accurate because there is movement of carbon from soil to groundwater, but my guess is it's not an appreciable amount given all of the size of all of the other fluxes. You're much more likely to see litter fall and things like this flowing into surface waters than you are to get significant carbon in groundwater. Am I misrepresenting that, Nathan?
No, that's correct. It's part of the slow cycle. I mean, the groundwater moves so slowly that it can get saturated with a little bit of carbon, and it takes decades for it to come out.
Yeah, and generally when you look at groundwater, groundwater is known for being pretty clean. And if there was a lot of carbon in groundwater, that would serve as an energy source for organisms, and you would actually see lower water quality in places with a lot of carbon in the water.
Yeah, it's just dissolved CO2. It's not a bunch of organic carbon in there as well.
Yeah, yeah. So what else is in this figure that was not in our simpler figure on the board?
Volatilization.
Volatilization. What is that, Devin?
Don't know what it is, but I just know it wasn't in the last thing.
Okay, so you'll see that it's associated here with fire. Volatilization is what we use to describe when chemical compounds are broken down and released through the process of burning biomass. And so when fire affects Konza Prairie, you're going to have carbon dioxide released back to the atmosphere because that glucose that is in plants is being broken down. But rather than being broken down in a controlled reaction that is governed by enzymes, it's being broken down by fire, much more rapid and chaotic chemical reaction than a chemical reaction that is moderated by the action of the enzymes. So I think that shows you all of the fluxes. But what I want to talk to you now and want to bring attention to you now is the actual numbers. So when we look at the atmosphere here, it says there's 760 times 10 to the 15th grams of carbon in the atmosphere. And then every year, about 120 times 10 to the 15th grams of carbon per year move from the atmosphere to the plant community through the process of photosynthesis. And then when you gather up all of the cellular respiration going on by plants and animals, primarily on the surface of the land, going back to the atmosphere through cellular respiration is about 60 times 10 to the 15th grams of carbon per year. And then these organisms ultimately die over the course of a year, and they go into the soil, and the bacteria in the soil use those glucose molecules for energy, and they return about 55 times 10 to the 15th grams of carbon per year to the atmosphere. So what happens when you do this math is we have 120 times 10 to the 15th grams of carbon per year coming into the ecosystem. We have 60 going out from cellular respiration, and then we have 55 leaving the ecosystem through soil respiration, which basically means that we have 5 times 10 to the 15th grams of carbon per year that stays where?
It stays in the soil.
It stays in the soil. So this is a cycle that would normally store carbon in soil. So it's an imbalanced nutrient cycle because more of that of this element is coming out of the atmosphere into the ecosystem, then is being put back into the ecosystem. And so it's imbalanced such that ecosystems would typically store carbon. But one of the problems is we have increasing levels of carbon dioxide over time, and this becomes a problem because carbon dioxide is a greenhouse gas, and as we get more and more carbon dioxide in the atmosphere heats up. And so people who are concerned about global warming are concerned about carbon dioxide emissions. But we just looked at this and we said, well, this system should be storing carbon in soil. Why is there not a net storage of carbon in this cycle? What other things does this not account for?
Fossil fuels?
What's that, Tessa?
Is it fossil fuels?
Okay, so one of the things that happens is humans dig up fossil fuels and put fossil fuels into the atmosphere. That ends up being about 5.3 times 10 to the 15th grams of carbon per year. And so just by burning fossil fuels, we now take 5.3 more and eject that into the atmosphere. So now we're exporting to the atmosphere about 0.3 times 10 to the 15 grams of carbon. What else happens that takes this and converts it from a cycle that would normally store carbon to a cycle that exports carbon to the atmosphere?
Volatilization.
Volatilization. So whenever we convert forests to farmland, in addition to the normal cellular respiration that's being done by plants, when we burn those plants up to clear land for agriculture or shopping malls or whatever else, we add another 3.8 times 10 to the 15 grams of carbon per year to the atmosphere once again, which gives us 4.1 times 10 to the 15th grams of carbon per year that goes back to the atmosphere, such that instead of the carbon cycle storing carbon in terrestrial ecosystems, we actually have a carbon cycle where we're putting more carbon back into the atmosphere. And that difference between storage in terrestrial ecosystems and a buildup of carbon dioxide in the atmosphere is all about human activities, fossil fuel consumption, and land use change, land use, land cover change. And otherwise, we would just be storing up carbon in soils. And rather than this chart of carbon dioxide increase in the atmosphere, we would actually see this thing going down. We're out of time now, but I want you between now and Wednesday to take a look at this figure of carbon dioxide, and you'll see that it's going up, but it also oscillates back and forth. And I want you to think about what the explanation might be for that oscillation. There's a good biological explanation for it, and we'll begin there on Wednesday. Questions?
Do you know if lab will be in person tomorrow, or because we're still technically C right now?
We're not in C. We've been released from C, so I will plan on meeting with you in the lab spaces so that I can actually look at your computers and things like this. But if for some reason that you are not able to meet because you're in quarantine, drop me an email and I'll get you logged onto Zoom so we can interact with you with your group.
Perfect. Thank you.
Sure, no problem. All right, I'll see you guys on Wednesday, if not tomorrow. Nathan, we're behind.
Yeah,
So-
I took an extra day for photosynthesis, basically.
Yeah, what do you want me to do about that then?
Well, let's see. I haven't really thought too carefully about how to recover from that.
Oh, crud. Were we recording that? Oh, yeah, we were recording that.